Hey everybody, Jeffrey Scott here, Meridian Biosolutions. Thanks once again for checking in with us. I certainly appreciate that. Don't forget to follow us here on Facebook. Follow us on YouTube as well, so you get the latest updates and all the cool information we're putting together, all about regenerative medicine. So if you've looked at the videos here on our website or on our YouTube page, we're starting to talk a lot about the importance of cellular senescence. So I want to take a moment and just say, or just address what are triggers for cellular senescence. And I think what I want to start with is the concept that cells have a finite ability to divide. Okay? And the reason for that is there are components called telomeres that cover the ends of your DNA, right? Your DNA is a double-stranded helix, okay? So on each end, kind of like that little plastic piece at the end of your shoelace, right? And if you've ever had that rip off, what happens when it rips off or a little bit rips? Well, it doesn't hold that lace together quite as nicely. Right? It may start to fray a little bit. And it's a similar type of phenomenon with telomeres. It's called telomere shortening. And once they get short enough, and this could be anywhere from 50 replicative cycles to 75, 80, 90 replicative cycles, depending on the particular cell type. But once it gets to that point and they get short enough, they're not doing their job properly with that DNA. And the ends of those, that double helix can get a little sticky. There can be some cross-reactivity, with other genetic material in the nucleus, which is going to cause problems. There may be issues with fidelity of transcription because those telomeres just aren't performing the way they need to perform. So when those get to a certain level, and in some cases you may even have DNA strand breakage, and when that stuff occurs, the cell senses that and turns off, and it stops its own ability to replicate and pushes itself into a state of senescence. Okay? Now, when it's in that state, that triggers, first of all, that's a good thing. Senescence actually does some good things. We talk about it in the context of aging as being a very bad thing, and it can be. But it has evolved to be a good thing in fighting against, for example, let's say you had a gene mutate. There are pathways that sense that and can trigger a biochemical pathway that moves that cell into senescence to prevent that one individual cell from becoming tumorigenic, okay? So that's one trigger. Oxidative stresses can push a cell into senescence as well, and it's rare that you're going to have one of these by itself. Oftentimes it's a combination of both of these type of factors, but oxidative stresses can push cells into senescence. So maintaining your state of antioxidant capability is very important as well. So by and large, those are the two main triggers, and I should say three. I did bring up the mutation. So telomere shortening, oxidative stresses, transformation of a gene or a mutation in a gene that could potentially be lead to tumorigenesis. Our cells are smart enough, they're amazing. They understand that that's not a good thing. So that's by and large, that is how cells are triggered and moved into a senescent pathway. And then from that point, it gets much more complex with some good, bad, and ugly consequences, which as we move forward, we'll talk about all of those in more detail. So thank you for listening. Don't forget, follow us here, YouTube, check out the website, Thanks for listening. Stay regenerative, my friends.